This technique can help answer key questions in the immunology field, such as determining how monocyte subset proportions or expression of different markers are altered in disease states. The main advantage of this technique is that the cells are near to their native state and that clear instructions and justification are used to provide a standardized gating method.
Generally, individuals new to this method may struggle because many papers do not give clear instructions on how gating of the cells was performed.
Begin this protocol with collection of blood samples and determination of the white blood cell count as detailed in the text protocol. Dilute the blood with phosphate-buffered saline to adjust the concentration to approximately 5 million white blood cells per milliliter. Prepare sufficient master mix for the number of tubes by combining blood, anti-CD14 V450, anti-CD16 APC, and anti-HLA DR per CP. Vortex and pipette 51.9 microliters of mix into each tube. Add PE-labeled M1 and M2 marker antibodies, as well as markers for T cells, B cells, neutrophils, and natural killer cells to their corresponding tubes. Vortex and incubate for 30 minutes at 4 degrees Celsius in the dark. Now, add 250 microliters of combined red blood cell lysis and white blood cell fixing solution and immediately vortex the mixture gently. Incubate the vortexed mixture for 10 minutes in the dark at 4 degrees Celsius. After 10 minutes, add 250 microliters of PBS and spin down the cells at 260 G for 10 minutes at room temperature.